Hey everyone. So after learning about DNA and its structure, we're now going to learn about some other types of nucleic acids called RNA and how they play a role in the production of proteins in our cells. Now your cell does this through two processes called transcription and translation, which we'll look at more in depth and what your cells actually do to produce these proteins. So you've learned earlier this year that the nucleus controls everything that happens inside of the cell. And that's because inside of the nucleus is our DNA. DNA contains the information that is needed to build proteins. It's kind of like the recipe that your cell uses to build these proteins, or instructions. So the problem is that DNA normally doesn't leave the nucleus. It's too big, too valuable to risk being damaged, so instead it's going to stay inside of the nucleus. This is where RNA comes in. Now looking at RNA, you can see two diagrams here, one of DNA and one of RNA. DNA is that classic double helix structure that we learned about last chapter. RNA, on the other hand, is a single-stranded structure. And you can see it's basically like that twisted ladder that only has one side to it. That's one of the major differences between these two molecules. Here we can see a little more clearly that RNA, again, is single-stranded. And RNA is a nucleic acid that is used by the cell in the process of protein synthesis. If you take a look in the upper left, you can see the nitrogenous bases that make up the RNA strand. Remember that the monomer of nucleic acids are nucleotides, and each nucleotide contains a phosphate, a sugar, and a base. You guys learned that last unit in DNA. If you look at the bases that RNA uses, you'll recognize three of them. Adenine, guanine, and cytosine were bases that you saw were also used in DNA. What you'll notice is that the base thymine is missing, and instead RNA replaces it with a base called uracil. So that's the second difference between RNA and DNA. After already going over the first two differences in previous slides, the third difference is that RNA, instead of using the sugar deoxyribose, uses the sugar ribose instead. It has a slightly different structure, but as far as you guys need to know, you just need to know that it uses ribose sugar instead. As we said, RNA plays a few different roles in the production of proteins, and you'll see here that there are three main different types of RNA that help the process. The first is mRNA. mRNA is called messenger RNA, and basically it contains all of the information needed to build the protein. It's going to be the copy of the code from DNA that then can leave the nucleus to the cytoplasm where the proteins are actually going to be made. rRNA, called ribosomal RNA, actually makes up the ribosome. You guys learned earlier this year that ribosomes were structures in cells that help to produce proteins. And that's true. We're going to learn exactly how they play a role in this unit. What you didn't know is that ribosomes are actually made up of RNA. And this RNA will be used to read the mRNA and to basically put the correct amino acids in the correct order. Lastly, we have tRNA, also known as transfer RNA. These RNA molecules are going to be responsible for carrying amino acids to the ribosome in the appropriate manner to make sure that we build the protein correctly. So now that we've introduced the differences between DNA and RNA, we've also introduced three different types of RNA that are going to play a role in the production of proteins. We're now going to transition to talk about the actual process, how these proteins are made. If you remember from earlier in the discussion, there are two processes that take place in order to produce proteins, and those are transcription and translation. Transcription is the first process that takes place, and it takes place inside of the nucleus of the cell. Here, DNA is copied onto messenger RNA using the base pairings G to C and A to U instead of thymine. So adenine, which normally would have paired with thymine, thinking back to DNA, now pairs with uracil instead. This process is called transcription. Let's try one of these problems with transcription out. So we're going to convert this DNA strand into a strand of mRNA. So the DNA strand reads TTG, GCC, AA. And we're going to transfer that into mRNA. So why don't you take a minute and try to do that. So using our base pairings that we learned last unit, you'll see that T pairs with A, so the first two letters would be AA. G pairs with C, so the third and fourth letters would be CC. C pairs with G, as we just said, so the fifth and sixth letters would be GG. And A pairs with U, so the last two letters would be UU. Again, we're using those same base pairings that we used when learning about DNA in our last unit. The only exception is going to be replacing those thymines with uracil. Now the process of transcription is a little more complicated than we're making it here for you. There are enzymes involved in unwinding the DNA so that it can be read from one side to another, and it's a more complicated process than we're showing you here today. But at the end of it, when we've created our mRNA strand, that mRNA strand will then leave the nucleus through those nuclear pores that you guys learned about earlier this year and travel outside of the nucleus into the cytoplasm. That's going to be where it's going to look for a ribosome, which will be the site of where these proteins will be made. Alright guys, so that wraps it up for transcription. Remember, we talked about introducing the differences between DNA and RNA, those three major differences. We also talked about three types of RNA that are going to play a role in the production of proteins. And we talked about transcription and how your cell takes DNA and converts it into a strand of mRNA where it can then leave the nucleus and create a protein outside in the cytoplasm. All right, thanks guys.